I warmly welcome all of you to our monthly webinar series conducted by the Center for Plant Materials and Herbal Products Research, University of Sri Jayewardenepura, in collaboration with the SLAAS Section D, Sri Lanka Association for the Advancement of Science. And today, as the resource person, we have Dr. Gayani Nanayakkara. She will be delivering her speech on cell-based assays. Dr. Gayani has received her PhD from Auburn University and then her BSc Honours degree in Pharmacy from University of Colombo. And currently she's working as a research associate at Eccles Institute of Human Genetics, University of Utah. Over to you, Dr. Gayani.
Thank you so much for the wonderful introduction, Dr. Munasinghe. So I'm going to share my presentation with you all. Good evening, everyone. So today I'm going to talk about cell-based assays. Before I start, I would like to thank Bhanuki Jayasuriya, who invited me to present today in this seminar series. So Bhanuki was one of my junior colleagues when I was in University of Colombo. From the flyer that was distributed, I gather that all of you are interested in plant material and herbal product research. But I have to declare first that I don't have experience working with plant material before, but I do have extensive experience working with different cell cultures. So today what I'm going to do is focus how you can cell cultures for drug discovery and development. So the outline of my talk would be, first I would describe why we do cell culture based assays. And as I was told that most of the audience would be undergraduates, I thought that I would give a brief explanation about the various cell culture types that are available and the cell culture conditions. And today I'm going to talk only about cell viability assays due to time constraints, and also I didn't want to make a very dense presentation today. And finally, I will talk about pros and cons of doing cell culture. So as most of the audience are undergraduates, and I was thinking that they might have plans to go for postgraduate studies, I thought I would start with talking about basic research method. So if you go for graduate studies and after you complete your graduate studies, if you're planning to do a postdoctoral fellowship, so most of your time will be spent on doing research. So as you all know, so the research method starts with developing a hypothesis. So you have to have some prior knowledge about the research area that you are interested in. So this prior knowledge may come from the data that your lab has already collected, and also by the publications that are published by other scientists. So after extensive literature survey, you can develop your hypothesis. And to test your hypothesis, you have to design a study. And when you conduct the experiments on these studies, you can collect data, and based on the data, you make conclusions. And your conclusion might be that your hypothesis is right or wrong. So at the end of your study, you have to make a scientific story and publish that story in a peer-reviewed journal. So when you design your research project, you have to really consider about the biological relevance, especially when you do a study design. So biological relevance means you have to use appropriate models to test your hypothesis. For an example, if you have a drug molecule that you believe is beneficial in Alzheimer's disease, so to test that drug, you have to use appropriate cell cultures like a neuronal cell culture. So it's no point testing such a drug on a liver cell culture, right? So you have your study has to be designed carefully, and it has to biologically really relevant to answer the questions that you raised in your hypothesis. And the other place that the biological relevance comes important is when you go to publish your work. So usually you publish your work in peer-reviewed journals. So once you publish your manuscript, the external reviewers will assess the biological relevance of your story. And so what I'm trying to tell is, like most of the time, if you collect data only from cell culture work, sometimes it's difficult to publish your story in journals with a high impact factor. So most of the time, the reviewers might ask for appropriate animal model data, that the data you have collected from the animal models as well. So there are various models that you can use for your study. You can use biochemical assays, for an example, like you can extract proteins from DNA recombinant technology and do the experiments in a test tube. And also you can use cell-based assays using cell lysates. In here, what you would do is mostly if you are testing a drug, you will grow your cells, put the drug into the cells, and then you will take the cell lysates and then do the experiments like maybe Western blots or immunoprecipitations. And also you can use cell-based assays using live cells. So most of the assays that I'm talking today are done on live cells. And also you can use animal models. So you can see when you go from sort of left to right, the biological relevance increases. Then the question comes, right? So if the animal models are the most biological relevant models for your study, why are we even talking about cell-based assays? That is because if you are going to do high throughput screenings, like if you have about 50 to 100 drug molecules that you want to test, you can't do that on animal models because of these reasons. Animal models, if you're working on animals, it can be really expensive, and it is not cost effective to test about 100 drug compounds on animals. And also working with animals can be labor intensive and also time consuming. And also, like if you have about 50 drug compounds, and most of the time, most of the drug compounds might not work, right? So eventually you will end up with 1 to 2 potential drug candidates. So if you test the rest of the 48 ineffective drug compounds on animals, and these animals sacrifice their life for nothing, right? So it's sort of unethical. And usually if you are working with animal models, your study has to be approved by the university. And also if you're funded by the US government grants, the government, you have to justify to the government why you are using so and so animal models and the numbers that you're using for the study. Because of that, you don't do high throughput screenings on animals. For that, you do cell culture, you do in vitro assays, and you test your 50 or 100 drug compounds. And then you select 1 or 2 potential drug candidates, and then you only use those 2 or 3 drug compounds that you use and test it on animals. So that's why cell culture becomes so important in drug discovery and development. So typical applications of cell-based assays when it comes to drug discovery and development, you can do the effects of your drug and see how it impacts the cell proliferation, and you can use cells to determine the toxicity of your drug compound, and also you can use migration assays, and you can determine what cell signaling pathways are activated by your drug. So you can use cell culture assays to assess all these. And when it comes to systems of cell culture, there are 2 main types. One is adherent cells and the other one is suspension cells. So most of the mammalian cells that you might work with our adherent cells. So the adherent cells, they adhere to the bottom of the culture vessel. So they don't freely, circulate in the medium. So one important sorry term that comes with adherent cells is confluency. So confluency means, like, if you look at your culture dish under a microscope, and you visually determine what is the surface area that is covered by the cells. Like here it says 10%. That means 10% of the culture vessel are covered by cells. But when you let these cells go over time, they will grow and they will replicate, and with time, the confluency of the cells will increase. And usually when it comes to adherent cells, and if you are planning to passage these cells for a long time, you just don't allow them to go above 80% confluency. That is because most of the time when the cells become too confluent, their gene profile might change, and also there might be epigenetic changes that you don't want to happen. So when the cells reach about 70 to 80% confluency, you have to passage the cells, right? So passaging means, like, when your cell culture plate becomes crowded, you take the cells and split them into several plates so they get more space to grow. Like, for an example, let's say that there is a classroom filled with students, right? And these students do not have even a place to sit. That is a highly unproductive classroom, right? So what you do is you take the students, split them into groups, and maybe put them into 2 or 3 classes. So then everybody has space to work. So that is a similar concept. When the cells become too confluent, you have to passage them, and then you have to put them into new plates so they get more space to grow. And because the adherent cells attach to the bottom of the plate through protein attachments, when you want to passage them, you sort of have to use enzymes like trypsin to digest off those protein attachments and lift the cells. So you require enzymatic digestion for adherent cells when you want to passage them. And also you need tissue culture treated culture vessels. So so that is done by the manufacturer mostly. So they sort of put a positive charge at the bottom of the plate, so it's easy for the adherent cells to adhere to the bottom of the culture vessel. And suspension cells may not be as common as adherent cells, but if you take cells of hematopoietic origins like monocytes, T cells, they are mostly suspension cells. And their growth also limited by the concentration of cells in the media. So because of that, suspension cells also need periodic passaging. But the thing is, here you don't need enzymes because they don't attach to the plate, and they don't need tissue culture treated culture vessels. So there are different cell culture types available, mostly the primary cells, which can be directly isolated from the animal tissues. So imagine, like, sometimes you have a tissue banks, right, where human donors donate their organs. So sometimes you may able to get human tissues. So if you get a piece of human tissue, maybe from heart tissue, so what you can do is you can take a small piece, cut it into small pieces, and subject that small piece of tissues for enzymatic digestion. So most of the time, the common enzyme that is used for primary cell extraction is collagenase. And the thing is, when you subject the tissues for enzymatic digestion, most of the time you get a heterogeneous mixture of cells. Because as you know, right, an organ is comprised of various types of tissues. So if you if you take a heart tissue and digest it, most of the time you end up with cardiomyocytes and fibroblasts both. So if you want to concentrate cardiomyocytes only, so there are protocols that are available. Like, what you do is you take the cell suspension and put it onto a large flask, and let it stand undisturbed for maybe about 15 minutes, because the fibroblasts attach to the cell bottom very quickly. So what you do is you let the fibroblasts attach first and take the cell suspension out, which is rich with cardiomyocytes. And once you get your cells, you can culture them and do your experiments. There's a disadvantage of primary cells. It has limited lifespan. You can't keep on passaging primary cells forever, because these cells undergo senescence and they just stop growing. Senescence means they age. So once they age, they stop growing, they stop replicating. One advantage of primary cells is most of the time they will mimic the normal physiology, because you extract the cells from tissues. And also another disadvantage of primary cells is the properties of the cell might differ from donor to donor. So that is a disadvantage of primary cells. Most of the time, most of the primary cells can be bought from a vendor. So the vendor usually checks the properties of the cells before they sell it to you. So so if you buy your primary cells from a company, usually you don't have to worry a lot about a donor to donor difference, because they test it before they send it to you. And there's another type of cells, secondary cells. So secondary cells are derived from primary cells, and they are usually subjected to a process called transformation. And some primary cells can undergo transformation naturally, or humans can induce transformation virally or genetically. Transformation simply means you manipulate the cells so it will override the cell cycle. So these primary cells, so when the primary cells are transformed, they get converted to secondary cells, which we also call immortal cells. So unlike primary cells, secondary cells can, replicate for generations and generations. So here I have given examples of most of the common immortal cell lines. One is HeLa cells, which was derived from African American woman who was suffering from cervical cancer. So it is more like a cancer cell line. And the name of this African American lady was Henrietta Lacks. So this name HeLa came as an honor to her. So these HeLa cells has been, used extensively in cell culture work, and also there is HEC293 cells, which are commonly used and which is very easy to work with. And these cells were extracted from a human fetus, and they are of kidney origin. And also there are Jurkat cells, which were extracted initially from a patient who was having T-cell leukemia. So you can see, right, the HeLa cells and the HEC293s, they are more like adherent cells, while Jurkat cells, they are cell suspensions. And so because most of the audience are going to be students, I thought I would briefly talk about the cell culture flow chart. So if you want to propagate the cells, first you have to start with frozen cell vial. So usually the cells are frozen in vials called cryovials. If you buy the cells from a company, usually they will send it to you in dry ice, which has a temperature of -80 Celsius. And if you have cell stocks in the lab, most of the time they will be stored in liquid nitrogen. So when you want to propagate your cells, the first thing you do is you take your cryopreserved cells and you thaw it, and you put the cells with growth media into a culture vessel. Most of the time, when you want to propagate, initially you use T75 flasks. So 75 stands for the surface area of the flask. So T75 has a surface area of 75 square centimeters. So what you do is you put the cells with growth media into the culture vessel, and you let it stand undisturbed for at least 24 hours. And then on the next day, you will come and observe the cells under a microscope and see whether they have the proper morphology that you want them to have. And so most of the time, one T cell flask is not enough for your experiments. So then what you have to do is, once this T75 flask reach about 70 to 80% confluency, you have to subculture them. You passage these cells and put the cells into new culture vessels. So you will get more and more cells that are required for your experiment. So once you have enough cells, you can plate them for your experiments. And also you can have one maintenance flask. That is, if you're planning to do continuous cell culture based experiments, you don't have to take your cryo stocks always and thaw it out and subculture them, because this process takes about at least 2 weeks. But if you have a maintenance line going on, so you always have cells in hand and you can split them and do your experiments. But if you're not planning to do any experiments in near future, you can freeze them. So when you're freezing the cells also, you have to be careful. You have to use a special media called freezing media, and usually we use these coolers, which will freeze the cells very slowly over a long period of time. That is very important because right, our cells or the animal cells, the 70% of the cells are made of water. So if you rapidly freeze them, these water in the cells will crystallize very fast, and that will damage the cell structure and that will kill the cells. So it's good if you use a cooler to freeze down your cells. And usually what we do is we put the cells into a cryo vial like this and put it into a cooler and keep it in -80 freezer maybe for about 24 hours. Then if we want to stock the cells for a long term, we will transfer the cells into liquid nitrogen. So also when you are working with cells, you have to maintain proper culture conditions. That is very, very important when it comes to cell-based assays. When you are starting to work with a new cell line, it is essential that you go through the cell protocols and get familiarized with all the conditions that the cells require. So, and different cells require different types of growth media. So the purpose of adding a growth media to the cells is it provides all the nutrients required for the cell growth. Like it may have glucose, it may have amino acids, essential fatty acids for the cells to grow. And usually, most of the growth medium have phenol red. But some of the assays like which you're planning to do might get affected if the phenol red is present in the media. So if you're planning to do that type of assay, you can buy media without phenol red. So this is a media without phenol red. So in such condition, what you do is you propagate the cells with growth media that has phenol red. But when you are plating the cells for experiment, you will use this clear media. And also this growth media has to be supplemented with a serum most of the time. And the universal serum is fetal bovine serum. And there are special types of cells that you may need to supplement with bovine serum or horse serum. So the purpose of adding serum is it provides essential growth factors and hormones that are required for cell growth. And usually, depending on the cell, the serum concentration in the media can range from 2 to 10%. This is something I thought to talk about today. So sometimes, like depending on the cell might require a differentiation media. Not all, you don't need this for all the cells, only a certain cell types may require this. And so differentiation media is you take the growth media and add drugs or substances that induce differentiation of the cells. Like for an example, there is a mouse fibroblasts called 3T3L1. So once they reach 100% confluency, if you add insulin and dexamethasone into the growth media, after 7 days, they will differentiate into adipocytes. So, so adding insulin and dexamethasone into the growth media, that will induce these fibroblasts differentiate into adipocytes. And if you are planning to test any drugs on adipocytes, this is one way you can get adipocytes from fibroblasts. It's actually really cool to see like all these lipid droplets getting accumulated in the cells, and they look totally different after 7 days. And also when you're working with mammalian cell culture, you have to give them proper culture conditions. So usually we use carbon dioxide incubators. So carbon dioxide is not essential for mammalian cell growth. So the purpose of incorporating carbon dioxide into the environment of the incubator is just to control the pH of the growth media. And also you have to have optimum temperature for the cells, which is 37 Celsius. And also you have to provide adequate humidity inside the incubator environment, because if you don't maintain humidity in your incubator, if the environment is dry, that will make all your growth media to evaporate. So what you do is usually we put a tray of autoclaved water inside the incubator, so that will provide adequate humidity that will prevent the media being evaporated inside the incubator. And also, we all know this, right? So when we are working with cells, we have to have aseptic environment. We have to use a laminar flow cabinet. And the reason that I use this particular picture is I wanted to show you how much clutter that is present inside this cabinet. This is not something that I would like to do. I would like to keep less stuff in the cabinet when I work as much as possible. The thing is, like in a laminar flow cabinet, there are like air filters here, and also there are some air filters here. So if you keep too many stuff in your cabinet, that will obstruct the airflow, and that might prevent you having a proper aseptic environment for your cell culture work inside the cabinet. And obviously, when you work with cell culture, you have to use gloves. And it's good if you wear a lab coat as well. And also the other accessories like tubes, pipette tips, glass pasteur pipettes, you have to make sure that they are autoclaved. And also, you need 70% ethanol when you are working with cells, because whatever the things that you are going to put inside the cabinet, you have to spray them with ethanol first and wipe them and disinfect them. Then only you can put them inside the laminar flow cabinet. So with this, after this, I'm going to talk about cell viability assays. So cell viability assays measure how healthy your cells are. So why are the cell viability assays are important? Especially in drug discovery, they are very important, because if you, let's say you come up with different new compounds that you believe there can be potential drugs in the future. First thing you have to do is you have to test them whether these chemicals are toxic to the mammalian cells, right? So, so what you can do is you can grow cells and put these compounds and test whether these compounds are actually toxic to the cell. If they are very toxic, then there is no point doing studies on those compounds anymore. The other advantage is if you're working with brand new drug molecules, you don't know the correct concentration that you can use on the cells. So you can use cell viability assays to determine what concentration to use for your experiments. Like you can design an assay, you can have a very low concentration, like maybe 100 nanomoles to start with. and you can gradually increase the drug concentration, maybe 10 micromolar, 20 micromolar, 50 micromolar. And you treat with your cells with different concentrations, and you assess the cell viability at each concentration. So if you design an experiment like that, you can determine at what concentration that compound becomes toxic to your cells. And by that, you can optimize the drug concentration that you can use to treat your cells and get the data. One of the very basic cell viability assays, one of the earliest methods that was used to test the cell viability is tryptophan blue exclusion assay. So you will see most of the cell viability assays that I'm talking today based on this principle that the viable cells have intact cell membrane. So you all know that cell membranes are semi-permeable, so they don't allow everything or anything to come inside the cells, right? They are very selective. So tryptophan blue is a dye which cannot penetrate if the cells has an unbroken intact cell membrane. So the cells can exclude tryptophan blue. So if you add tryptophan blue, the live cells will not get stained because the dye cannot penetrate into the cell, but the dead cells get stained because they have their cell membranes are compromised. One of the disadvantages of tryptophan blue assays is you have to have your cells in a suspension. So if you are working with adherent cells, so the cells that attach to the bottom of the plate, you can't directly use this assay on those cells. So what you have to do is you have to put trypsin enzyme, digest the protein attachments of the cells into the cell culture plate, lift the cells up, make a cell suspension, and then add tryptophan blue. Usually, if I remember correctly, you usually add tryptophan blue at a 4% concentration. So what happens is, so when you add tryptophan blue, as I mentioned before, the dead cells will be stained blue. So once you have a cell suspension like this, then what you have to do is you have to count. You have to count the total number of cells present in your sample. And also you have to count the number of stained cells. How do you count them? You can do that manually by a hematocytometer. I'm sure all of you know what a hematocytometer is. So you can just put your cells into this hematocytometer and count the cells under a microscope. And your life would be much easier if you have automatic cell counters. So, so what you do is you present your data, like you count all the cells and you count the stained cells. So you can present your data as percentage of viable or dead cells, whatever the way you prefer. And also there's another method called MTT reduction assay, which is a simple colorimetric method. So this has a different principle. It does not rely on having intact cell membranes. What this dye relies on is the metabolic activity of the cells. For an example, so MTT dye, this MTT dye has a long name, which I didn't even bother to put it in the slide. So what you have to understand is here, the MTT as a dye can easily penetrate the cells. It's not like tryptophan blue. So MTT can easily penetrate the cells. The thing is, if the cell is alive and if it has mitochondria that is working, this MTT dye will be reduced into a purple color formazan. So this purple color compound only be produced in the viable cells only. So what you can do is, if you want to assess your drug, you can culture your cells, then you can treat your cells with the drug, maybe leave it for 24 hours. Then you can add MTT dye and incubate the cells with the MTT dye maybe for about 4 hours. And that if the cells are alive, this MTT will be converted to a purple color formazan, where you can detect it by measuring the absorbance using a plate reader. So the purple color that is developed will be directly proportional to the number of viable cells that you have in your culture plate. And there is advantages of MTT assay. The advantage is you can use it on adherent cells. There is no need to trypsinize them and make a cell suspension. And also you can optimize it to 96 well plate and can use a plate reader. So the advantage of having 96 well format is you can test multiple drug compounds at the same time, right? So because of that, it is ideal for high throughput studies. And there's another assay called risoserine reduction assay. It has the same principle of the MTT assay, but the only difference is MTT assay was a colorimetric assay, right? It measures the absorbance, and risoserine reduction assay is a fluorimetric assay. So it's the same concept. You treat your drugs, you treat your cells with the drug for however long you want. And then if you want to measure the viability after the treatment, what you do is you add risoserine, which can be easily penetrated into the cells. And if the cell is alive, this risoserine will be reduced to a compound called risorufine, which has high fluorescence capability. And what you do is, just like MTT assay, you use a plate reader and measure the fluorescence. Actually, you can measure the absorbance as well, but measuring the fluorescence is the more preferred method because it seems that the fluorescent data give more accurate data than the absorbance. So there is no much big difference between risoserine and MTT. So people say it's slightly sensitive than MTT, but depending on your study, we either use risoserine or MTT. I don't think that is going to make a big difference. Okay, so MTT and risoserine, they measure the cell viability. Also, you can do the opposite, right? You can detect the apoptosis. Like if you want to measure the cell toxicity of your drug compounds, either you can measure the cell viability or you can measure the apoptosis. So apoptosis is the cell death. So there are various players in the apoptotic signaling pathway, and one of the important family of enzymes are caspases. I don't know whether you have heard about caspases before. So in human cells, there are, I think about 12 caspases ranging from caspase 1 to caspase 12. And some of these caspases like caspase 3, which I'm talking here, and caspase 7, caspase 8, and caspase 9, they are more involved in the apoptotic cell signaling pathway, while caspases 1, 4, 5, and 11, they are more into triggering the inflammatory pathways in the cells. So imagine like, okay, imagine that you think you have a drug compound that has anti-cancer properties, right? You believe that that drug compound will kill the cancer cells. So if you have such a compound, what you can do is you can grow a cancer cell line and treat those cells with your drug and let it go for some time, and after the treatment is over, you can measure the apoptosis of the cells, right? If your drug was very effective, then most of the cells might be dead by the end of your experiment. So you can, so caspase 3 is one of the most important players when it comes to apoptosis. So caspase 3 is present in all our cells, but it is present as a proenzyme. Proenzyme means it is in an inactive state. So it's not going to do anything in the cell until it gets cleaved. So imagine in the previous example where you are testing an anti-cancer drug and where you expect that the drug will kill the cell. So once you treat the cells with the drug, it may trigger the cell death pathways and promote this caspase 3 in the cells to get cleaved. So it gets cleaved into P10 and P20 subunits, And once it gets cleaved, it can activate the downstream signaling pathways, and ultimately it will lead to breakdown of the DNA and cell death. So if you want to measure the apoptosis, you can use fluorescent-based methods. Like there are fluorescent probes that are available that can specifically go and bind to cleaved caspases, right? I mean, not only for caspase 3, like if you want to test caspase 7, caspase 8, caspase 9, and even caspase 1, you have specific peptide, fluorescent peptides that you can use for the caspases that you are interested in. And the thing is, when you add these peptides, fluorescent peptides, it will bind to the cleaved caspases, like here for the example, cleaved caspase 3. And during the washing steps, the unbound peptides will be washed off, and the peptides that are bound to caspase 3 will remain and they will fluoresce. So you can measure the fluorescence by a plate reader, or if you have a fluorescent microscope, you can use that to measure the fluorescence. Or if you have flow cytometer in your institute, you can use flow cytometry to assess how many cells are dead. So out of all these 3 techniques, I would prefer flow cytometer because you can assess individual cells using a flow cytometer. Like if you use a plate reader, if you use a microscope, usually it gives you the fluorescence of the whole cell population in the culture vessel. But if you use a flow cytometer, you can individually detect cells, right? So if you're using flow cytometer, like you can detect 50,000 or 100,000 cells in a matter of minutes. So out of all these 3 techniques, I would prefer flow cytometer for this assay. And another common way of detecting apoptosis are tunnel assays. This also based on the same principle where the cells with intact membranes do not allow certain compounds to penetrate. So propydium iodide is the dye used here. So this dye cannot penetrate into live cells, but it can penetrate into dead cells and stain DNA. So it will fluoresce once it bound to DNA, and you can use plate readers and microscopy to detect the fluorescence. And microscopy is commonly used. So if you are using microscopy with a tunnel assay, what you can do is you can use a nuclear stain like DAPI. It need not to be DAPI, you can use any nuclear stain. And then you do a tunnel staining. And then what you do is you observe them under the microscope and overlay this DAPI image and the tunnel image. So when you overlay the 2 images, if the DAPI stain and if the tunnel stain co-localize together, that indicates that particular cell is dead. And here, these arrows mark the dead cells, and this blue nucleus, where the red color is not co-localized, this indicates that these cells are alive. They are not dead. So there are various other cells, other viability assays that you can use. You can use lactate dehydrogen assays, calcium staining, which I'm not going to talk today. And there are other cell techniques also available where you can cell culture-based assays where you can use to test your drugs on. And that also I'm not going to talk today because as I mentioned before, I didn't want to make this presentation too dense. So the pros and cons of cell culture are the pros, as I mentioned before, they are more biologically relevant than biochemical assays, and also they are cost effective than live animal models, and also it's easy to reproduce the data using cell culture methods. And also for high throughput screening, cell culture methods are ideal. And the cons, so when you do assays like cell viability assays, so one of the disadvantages that I usually come across is these assays, they have multiple washing steps, right? So you put certain dyes into the cells and you have to wash off the extra dye present in the well, because otherwise you get a lot of background in your data. The problem is when you do multiple washing steps, you lose the cells, like especially adherent cells, because you put washing buffers, remove it and add washing buffer again. So the cells get exposed to a lot of mechanical stress, and that might make the cells to lift up from the bottom. And when you remove the wash buffers, they will be removed with the buffer. So the solution to overcome this is you can coat your culture vessels, like you can use gelatin or fibronectin or poly-D-lysine to coat the vessels before you plate the cells. So if you coat it, sometimes the cells may adhere more stronger to the cell culture vessel. So when you do multiple washing steps, you might not lose a lot of cells. And the other method you can do is this is not possible to do with all the cell viability assays, but you can, once you get your final readout, you can lyse the cells and measure the protein concentration. So the protein concentration is directly proportional to the number of cells present. So what you can do is you can get your maybe the colorimetric data and standardize it to the protein concentration. So that is one way to overcome that problem. And also, when it comes to cell culture, it's difficult to mimic exact physiological conditions because you only take one cell type into consideration. And also another disadvantage is if you keep on propagating cells, especially immortal cells, for generations and generations, it might alter the gene profile, and also it might induce epigenetic changes, which you don't want. So those are the disadvantages of cell culture. So with that, I come to conclusion of my presentation. I would like to thank you all for participating today. And I would like to thank Vanuki again for inviting me. I didn't talk about most of the other assays that we can use on cell culture. With this, if you have any questions, I would be happy to answer.
Yes, thank you very much, Dr. Gayani. It was a really nice presentation. I think it was very simple and we could understand it, especially for the students. You explained it very in simple terms. Thank you so much for that. And I am proud of you being a University of Colombo graduate, where you also graduated. And thank you so much for joining with us today. Yes, please, if you have any questions, please, it's time for you to ask.
Thank you so much.
You're welcome.
Dr. Gayani, may I ask a question? So for cryopreserve the cells, so rather than using liquid nitrogen, would you recommend using like, I don't know that is possible or not, like minus 150 freezer? Would you recommend that?
If you have a freezer like that, you can use it, because I think even if you take liquid nitrogen, I think the temperature is around 190, minus 190. But that's not a problem. Even you can easily store for like few months in minus 80, so 100. If you have a freezer like that, it should work. That's not a problem.
Okay, all right. Thank you.
Dr. Gayani, why do you use phenol red in the medium? Is it just for the visualization or?
It is usually used to detect the pH because phenol red is an indicator. So if your cell culture becomes too acidic, that phenol red will become yellow. So it's a clear indication that the pH is not right in your cell culture. And also if it becomes too basic, it will sort of get a purple color. But usually if the cells are too confluent, so the phenol red do become yellow, that's a sort of an indication that your cell culture is not healthy. So it might not be, useful to do any experiments on such cells. So,
okay, so as a pH indicator only.
yes.
Okay.
Yes.
Thank you very much.
Yeah.
Yes. Any other questions from the audience?
Dr. Gayani,
Mm-hmm.
it was a very amazing, very interesting presentation.
Thank you.
What I want to know is if any contamination occur in any culture medium, then how we are going to avoid that? That means if any bacteria or fungi grown in our cell culture medium, then how we are going to avoid or clean those cultures?
Usually, like even though you use aseptic environments, sometimes you can get a contamination. But if my cells get contaminated, I would stop using them. I would not try to save them. Because when there is a infection going on, there are a lot of changes that is happening in the cells, right? It will activate the immune responses, inflammatory responses in the cell. So usually if there is an infection, I wouldn't use those cells for my experiments. I would discard them and I would start a new batch. So one way you can avoid is like you can add an antibiotic into the growth medium, like a pen strep. Usually we do that when we culture the cells. So into the growth medium, we add antibiotic to prevent bacterial growth. But if your cells are contaminated, I would not recommend on using them.
Thank you very much, Dr. Gayani. And I have another question. Now, all the assays that you mentioned are basically to test the acute toxicity based on acute toxicity. Then, if we want to test any chronic toxicity, then how we are going to do that?
So the toxicity that these assays, you can, I mean, if you're planning to, like, if you want to test your drug and treat them maybe 2-3 days, still you can use these assays, right? So maybe you can plate your cells at a low confluency and then add your drug and let the cells go maybe 3-4 days. And once your assay is completed, still you can use these drug assays that I talked about to assess the viability and whether the drug triggers any apoptotic signaling pathways. Still, you can use these assays for long. I mean, even for long-term treatment. It's just that you have to plan your cell culture for that. Like you don't start with like a very high confluency if you're planning to keep your cells for a long time.
Yes, and Dr. Gayani, if we want to purchase any of these cell lines,
Yes.
what is the place you would suggest?
In the United States, we usually get the cells from a company called ATCC. I can send you the link for that. But I'm sure that they might be having branches in South Asia where you can get the cells from. And also there are other companies that do sell the cell lines, but usually my experience is always with ATCC.
They have
American tissue. Sorry?
So those are reliable ATCs.
Yes, ATCC. I have never had problems with them. And also, if you are buying cells for academia, academic work, they sell it at a discounted price as well. Yeah, I will forward you that website.
Okay, thank you very much, Dr. Gayani. Yes, any other questions from the audience? Okay, if there are no more questions, I think we are running out of time today. I would like to thank Dr. Gayani again for joining with us today. And I got to know that you were there in the university at the same time I was seen. And I would like to thank Dr. Bhanuki also for introducing her to me. And I'm really proud of you being there and helping us today. And I think you're going to continue this. We are going to have more sessions with you in future.
I would be happy to, yeah.
Yes, thanks a lot. And I would like to welcome all of you. Now, we are having these sessions every month on the last Monday at 6 p.m. And I'll be circulating the flyer through internet, like email, and we'll be putting it later in our website also. We are planning to do that, update the website. And I welcome all of you to join with us in the next month's session as well. Thank you very much.
Thank you. Thank you for giving me this opportunity. Bye.
Bye.